Hi, this is Jim Lindemann again, and we're on to topic number 8, the first cell and first multicellular object or whatever. This is really, you know, the deeper you get into this, the more and more you find out that there's a lot of stuff here that I could present. Hopefully at least some of this will identify some of the uniqueness that we have because of a creator, and some of the things that are really fascinating, as well as what would have to be contained within the first cell in order to make the first cell viable and able to reproduce. So we get into that area a little bit today. Okay, first of all, what part do I play in the great ecological cycle of life? You're the heavy. And then you have the two worms, and they come together and they keep on going until there's nothing left. And, well, we'll leave that as an existential question for you to figure out, at least in regard to creation and evolution. Anyway, life, a jump of fantastic dimensions. A few minutes in the presence of such an animated blob, the protocell, would hardly bolster one's faith in the harmony of nature. The initial impression would be one of utter chaos. Yet there is an order in this strange commotion. It is an invisible, elusive kind of order called life, a phenomenon which in some ways seems increasingly difficult to comprehend as we learn more and more about it. The process to thwart chemical natural tendency, that is, the natural tendency, if a body dies, is not then to continue life or to resurrect life, but rather to decay. Natural tendency of chemistry is toward equilibrium, not necessarily the way that you find it. Excuse me, that you find it in a cell. The thing is that this natural tendency to go, or this ability to go against natural tendency would have to be operating already in the primordial mix if life is going to begin at all. It is not as if somehow a cell gets encompassed and now life begins. What you have to say is basically, according to evolution theory, is that you have to have life in the great big slime, whatever, before cells even exist. Oops, that wasn't what I wanted to do. All right, here we go. There is one step in evolution that far outweighs the others in enormity. The step from macromolecules to cells. All other steps can be accounted for on theoretical grounds, if not correctly, at least elegantly. However, the macromolecule to cell transition is a jump of fantastic dimensions, which lies beyond the range of testable hypothesis. In this area, all is conjecture. The available facts provide no basis for the postulation that cells arose on this planet. There is no scientific evidence. And that's something that's kind of important, because there's so many people that think that, well, once the theory has solved the problem, then there is no more problem. But the problem is that no, they don't know anything really about life. They can tell you about it, but they can't tell you what it is. It just happens in evolution, kind of like the Big Bang. It just happens. There's nothing there, and all of a sudden there's the Big Bang, and suddenly there's mass. There's a huge, huge amount of mass where there was absolutely nothing there before. Time and personification. Given enough time, everything will happen. The annoying question is, why should it? What is there that says that it has to do anything? Why should amino acids form long chains? If the state that the molecules are in is efficient as it is, why would there want to be any change? Why do you have the idea that they're going to want to progress up the scale? Evolution supposedly is purposeless and random, and yet supposedly it's always upward, never downward, always
irreversible, only one direction, always smaller to bigger, always less to more complex, always less to more perfect, is not repeatable. Well, who chooses what is upward? You know, these are goal-oriented. What's more perfect? These are quality statements based upon an anticipated goal and implied ambition. Why should common chemistry want to go any further? The moment you use that kind of language, you have a godlike personification to elements that don't have any kind of even personality, much less the ability to think, to plan, to goal, to everything else like that. Rather than an asset, time is destructive. Biological matter wears out, breaks down, decomposes in a relatively short time span. How would a primordial soup molecule be retained long enough, perhaps for thousands of years or even hundreds of years, when there is no mechanism to preserve nor to replicate? You know, you have the sense is that once you have a molecule, the molecule is kind of put into safekeeping while we're working on others here. And that's not, there's no way that can possibly be true. Or if the molecule is going to be for a couple hundred years, maybe it'd be good to get a couple copies of it just to make sure that they last. But there's nothing in a primordial soup to guarantee that's going to happen. And yet, in time, everything that you got breaks down. Irreducible. This is we're going to embark upon a topic here that is really, really important, fascinating. Unlike beautifully formed snowflakes, which can be reduced to or explained in terms of ice crystal structure, no natural process can turn closely packed cubes of solid silver into coins. Looking for special environmental conditions would be futile, because the patterns are intelligence made. You know, in other words, you can have that sitting there for how many hundreds, thousands, millions, billions of years, and you're never going to even approach something like this, no matter how much time it's given. The coin structure is therefore irreducible to the physics and chemistry of silver. In other words, what you have here cannot be explained by that. And it was clearly imposed upon the silver by some intelligent external agent, in this case humans. Michael Poliani, in an article in Science called Life's Irreducible Structure, living organisms have a machine-like structure not explained by or reduced to merely the physics and chemistry of the molecules of which they consist. Therefore, life exhibits irreducible structure. In other words, you can have all the chemicals you want, but you're not going to have the kind of design that we see in nature simply because these things happen to hang around together. The structure and function of the parallel components in life cannot be reduced to the properties of carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, and trace elements that they are made of. Therefore, there are endless examples of such irreducible structures in living systems. That you can put all of this together, you can mix it together, you can do whatever you want to, and there is nothing that says that it's going to go any further than that. And you cannot have some of the most intricate types of machinery, if you will, that exists in the cell today. You get into a topic called autopoiesis, self-making. It comes from the Greek self and poieo, which means I make. It's a unique and amazing property of all life to continually repair, maintain, even reproduce itself using energy and raw materials from its environment. Now you take a look at a common cell, and this is just a representative of it, but you have all kinds of things between, nucleus, the nuclei, you've got ribosomes, you've got macrotubules, and all kinds of things that are there that, how do you explain them? You can't just simply have chemicals together, shake them together, and this is what's going to come out. there is such intricacy and there's such machinery that how do you explain it as simply as, well, it just happened? You have, say for instance, in a factory, and yes, you have all kinds of things happening, but the factory doesn't replicate itself. It's there that simply it makes things, and the things that it makes that don't replicate themselves. Contrast an allopoetic, Greek allo means other, entity such as a car factory uses energy, raw materials to produce an organized structure other than itself. In other words, this has to pre-exist its product before the product can even possibly show up out in the market. A hierarchy of irreducibly structured levels built upon but not explained by the level below it. And so you start out with the very basic, the components with perfectly pure composition. In other words, not only do you have the elements, but they are perfectly pure. You don't have a mix of things that are contaminating it. And these then come together into a highly specific structure. Now, they can't create the structure. However, the structure controls the elements. You go up to the next step, components that are functionally integrated. In other words, again, you can have all these pieces, but it does not mean that they're going to work together or come together. They're just going to sit there. I'll explain this a little bit more. This controls these things. They control, each level controls the ones below, but it cannot create the level above. Comprehensively regulated information-driven processes. Inversely causal meta-informational strategies for individuals and species survival. Okay, let's kind of put this in a little bit different. You start out with raw materials. The raw materials cannot create a design, like for instance, the coins, the silver coins. The raw materials will just sit there. Okay, so now you have a design. Now you have something that you're going to do with these raw materials. But you can end up with a whole bunch of shells, but you don't have any way that they're going to somehow fit together, that they're going to assemble the way they need to assemble in order to produce the outcome. And then once you have the thing assembled, it doesn't mean that it's going to work. You have to have something that's going to kind of switch it on and make it adapt to whatever it is that is in the environment. And then you're going to have some place where the information is stored, and preferably in a way that that information is checked and double-checked to make sure that it maintains its integrity. And the thing is that even then, where did this come from? It couldn't come from below, because the below doesn't have the capacity to do that. And so you're going to have something external in order to make that information available and then create the rest. An unbridgeable abyss between the dirty mass action natural environment chemistry and the perfectly pure single molecule precision of biochemistry. And we'll get into a little bit further, but again, even at this basic level, you have already a distinction between what is out there in nature and what is inside each of the cells. And it is quite a contrast between the two. We will talk about that when we get to purity. Okay, so let's take what we were just talking about and put this in terms of a vacuum cleaner. So a vacuum cleaner, when it first was put together, or first created, had transitioned from the basic motor housing exhaust fan, dust bag, and flexible hose to a cyclone filter and wheels, and then to small robotic machine that runs around all by itself until its battery runs out, to one which automatically senses battery rundown and returns to its housing to recharge. Okay, so what's going on here? For the vacuum cleaner to reach the living condition of autopoiesis, for example, It would need an onboard power generator using various organic, chemical, and light sources from the environment as raw material. In other words, it can't depend on anything other than what it can provide for itself. It would need a sensory and information processing system to determine the state of both the external and internal environments. So for the vacuum cleaner, how dirty is the floor? And what is the vacuum cleaner condition? Does its bag need to be checked? Oops. It would need to know where maintenance and where maintenance and repairs. So where does it go for maintenance, and where does it go if it has a screw loose? How does it fix itself? It has to know how to do that if it's going to be of that kind of condition that the cell is at in terms of being able to take care of itself, to self-make itself. All right. It would need to know where to expend effort, how to avoid hazards, how to obtain precisely required supplies from the environment, most often to make the required item from environmental raw material. So, okay, say for instance the aluminum housing on the vacuum gets cracked, or somehow gets a chip out of it. The vacuum cleaner would have to know where it should get the aluminum it's going to need. It's going to have kind of like a foundry in it to be able to purify the aluminum. It's going to have a processing by which it makes what has to be repaired, and then do the repair and do all of that. And it has to be able to know each step of the way. It would need the ability to replicate itself, not only to repair itself, but to be able to make a second one that's fully functional right from the word go. Although motor, housing, bag, wheels, and etc. are crucial, the mentioned abilities do not arise from such lower levels. In other words, if you have the housing, it's not going to make itself. It's not going to assemble itself. And after you have it assembled, how is it going to get started? How is it going to turn on? How is it going to adjust whatever it is to the rug, to the height of the nap in the rug, and all the rest? How is it going to know that unless you have some information that comes to it? It's not going to know that just because it's sitting on the rug. It's not going to know why and what it's about. Steps to autopoiesis must come from a higher level in an added design feature. Ultimately, there must be information from higher levels to control all lower levels and have the capacity to anticipate need, inverse causality, as well as meta-information, information about information. In other words, okay, well, we have this information, but how do we know what to do with this to protect it, to safeguard it? Information needed to preserve the information needed to stay alive and to ensure the survival and perpetuation of the species. It would need a programmer or programming center in order to get that information into the machine. So we're back to here. Raw materials, design, assembly, switches, computer, and programmer. All of this has to fit into that kind of a sequence where the vice does not create the next level up, but the next level up controls the lower levels. Inverse causality. Normal causality, cause and then effect. You know that you take a hammer and you hit a nail and it goes in, penetrates into the wood. There's the cause and there's the effect. Well, inverse causality, however, is the anticipation of the effect compels the cause. Okay, let's see if we can understand this. A condition now, anxiety or paranoia, is caused by an anticipated future event, perhaps harm to one's person. So, okay, let's take a look at cause and effect. Workplace hazards can exert direct toxic effects, normal causality. In other words, if you're working on this at a place that has high levels of asbestos, chances are you're going to breathe it in and it will create body problems for your body, ultimately perhaps cancer. But a dreaded potential harm can exert toxic effect. In other words, that if you have workers who know that there is asbestos in that atmosphere, in that workplace, and they refuse to go in because they don't want to become exposed, this is inverse causality. The anticipated result affects their choice or their cause at this point. Mark Twain once said, "I have suffered a great many things in my life, most of which never happened." In other words, the anticipation of what he thought was going to happen had so severely affected him that he, as he put it, he suffered a great deal, and basically discovered afterwards that those fears were groundless. And yet he would go in with anxiety, with worry, with all these other kinds of things in anticipation of a certain effect. Okay, certain behavior now, investment decision, is caused by an expected outcome, anticipated profit or loss. So in other words, do I buy? Do I sell? Do I hold? What should I do? Well, I kind of try to figure out what the future will happen, and then I will make my decision as to whether I will do any of those functions in terms of my investments, so that the anticipated future event causes my decision now. What's called self-fulfilling prophecies, expectations which influence the environment to create the effect. So in other words, you have somebody that says, nobody will like me. Well, and therefore the person is irritable and crude and nasty to everybody else, and by golly, nobody likes me. So look, I told you they wouldn't. But the thing is that the effect was created by a cause prior to it. The cause was that I acted as if they would be nasty to me, and that was the outcome. So you have inverse causality. It goes backwards. The result is what influences the cause or the decision, or however you wish to make it. So an embryo forms organs, for instance eyes, not based on present need, but on future need. It has no need for eyes at the moment. While it's in the womb, it really is not going to be seeing anything. But the thing is that the eyes are made so that when the time the birth comes, everything would be there as needed and functional. Evolutionists avoid this, admitting such things, because life then might suggest that it has purpose, that there is actual direction, there is actual reason why something will exist, rather than just simply that it happens. What will be is already programmed into the fertilized egg. You think of it as a nanobot, this very, very tiny thing. And it's got everything that it needs, everything in order to operate, everything to build a complete human being and make that human being able to handle its environment, along with necessary equipment to produce the machine. The egg has it all, ready to go. And so basically, you supply it with all that it needs, the nutrients, the environment, the whatever, and it will go from level to level to level, creating what it needs to finally end up with a complete human being. Okay, purity. Nothing less than 100% purity will work in the molecular machinery of the cell. You have chirility, left and right-handedness. You have enantiomers, molecules that can exist in both forms, mirror images. In other words, you can have left-handed amino acids and you can have right-handed amino acids. You can have left-handed sugars and you can have right-handed sugars. And they will exist in nature in both. Synthesis produces 50-50% mixture called a racemix, however you want to pronounce it. Homochirility, left-handed amino acids, right-handed sugars. In the cell, you only have amino acids that are left-handed. In the cell, you have sugars that are only right-handed. And one of the problems, one of the great tragedies of years past was that this was the issue, the pivot for what was called the thalidomide drug disaster. That instead of having left-handed amino acids, which was great for pregnant women, the company was not careful about the chirility of the amino acids. The other form caused the shocking fetal abnormalities. So in other words, this is so critical, so crucial, and this is governed so that in the cell there is no other alternative than what is required. Protein assembly in ribosomes illustrates single molecule precision. You have the DNA molecule's coded protein structure transcribed to messenger RNA. Transfer RNA brings the next required individual amino acid for the growing protein chain. You have everything assembled literally molecule by molecule. It is not as if you take the molecules and you throw them together and you hope that it's all going to turn out right. But instead what you have in the cell is very, very, very precise building of the elements that the cell needs in order to conduct its business. Adding one molecule at a time monitors and corrects even single errors, the result is extremely fast, perfectly pure chemistry. That these are things that are going on constantly in your body, and they're going on thousands of times in a minute. Outside the cell you have what's called dirty chemistry. Not because it's, not as a way of depreciating it, but simply because there's contaminants. They're always present. There's always a racemix, whatever. And so you always have that kind of danger out there. And the cell, as it's going to take things into it, has to be able to choose and to reject. The law of mass action in the test tube versus the cell's single molecular precision, controlling reaction rate, outcome through enzymes and nanoscale structures, pathways, and resulting often in a totally different biochemical reaction. In other words, you got things happening in a cell that would not normally happen. You have bases and acids which should react violently with each other, working inside a cell and co-working. Experiments typically produce muck, goo, and gunk. In other words, when you have outside the cell to try and replicate, you don't end up with something that is this orderly and precise. You end up with basically, well, really, what is a toxic mixture. Autopoiesis meta-information. The information and associated abilities needed to repair, maintain, and reproduce in perfect condition, needed to protect and pass on the information also in perfect condition of the characteristics and mechanics of life throughout its full existence, ensuring both the cell as well as its descendants' survival. In other words, you have the basic operating information, but then you have the information that governs that information and double checks it and makes sure that it is being properly unfolded. Some information activated in anticipation of a future need, in embryo's development of the eyes, which is inverse causality, or for a later stage of organism development. In other words, you have the information that is protected and waiting for the time when, say, for instance, puberty comes, for the body changes that are necessary at that particular time to equip the person for their function further on. Some information contains built-in resilience, what's called differential reproduction, to be able to handle environmental changes, So, like say for instance with the sperm and the ova being only half chromosomes, and now you've got two coming together, that there is a resilience that this provides to things that are happening out in the environment. Changes out there. Well, this is the way you get the fine-tuning, you get the adjustments that are already there in the DNA. It's just that now you can bring them out, or they now can come out and handle whatever is the need of the moment. The huge number of permutations and combinations to provide offspring with survival variations, meanwhile protecting essential processes of life, the ability to read the DNA code and translate it into metabolic action. So you have all of this information that's going on, being able to accomplish all of these kinds of things, and then you have the information that is the safeguard of that information. Nothing ever happens on its own in a cell. Something else always causes it, links with it, benefits, or is affected by it. All links are multi-step processes, so that it makes it harder and harder for evolution to be able to describe why you have this sequence. Because the sequence doesn't work if you only have half of it. We get to this when we get to ATP, adenosine triphosphate, the powerhouse, or the power for the cells. All links are choreographed, intricate and harmonious regulation by information which never occurs in a natural environment. DNA is an ideal information storage facility, but it cannot create information. The cell elements are ruled by information, but cannot create the information. Again, you have the lower level cannot create the upper level. The lower level cannot create the supervising information to make it work. As with the vacuum cleaner, the higher source of information is required until finally you get to a programmer outside of it. Irreducible functions. If life began in some stepwise manner without autopoiesis, then autopoiesis is the end product of a long and blind process of accidents. This is what evolution wants you to understand or believe. Therefore, some organisms should never have attained it, because after all, if it's something that acquired over time, some wouldn't acquire it. Some organisms should have lost it because they did not need it. If it was something that was acquired, then it wasn't needed right at the beginning. But what we have is that these kinds of things, these kinds of controls and oversights and everything else like that that go on in the cell have to be there right at the beginning, otherwise things wouldn't happen. Autopoiesis is universal in all forms of life, so it has to be essential. Experimental origin of life projects which begin without autopoiesis have proved universally futile in achieving even the first step towards life. In other words, if you don't have that kind of information that's going to safeguard and work the whole system through, it won't happen. Just the same as if you were to take all the raw elements and throw it all together, that the fact that they would combine and combine in such a way that would make the next step possible just doesn't happen. Each level of the autopoietic hierarchy is dependent upon the one below it, but is causally separated by a Polyanian possibility. Autopoiesis, therefore, cannot be reduced to any sequence of naturalistic causes. This is the big problem that evolution has to face. Autopoiesis is required at the beginning of life even to exist and perpetuate itself. In other words, the ability to replicate, the ability to repair, all of this. Where does it come from? It doesn't come simply because everything was thrown together and could not have turned up at the end of some long naturalistic process. Because if it didn't need it in the beginning, it certainly doesn't need it now. Why would it even show up? So, one of the problems with the first cell is that you have simultaneous structures that are required. You have to have all of these things in place, otherwise it's going to be a cell that just literally goes nowhere. First, you have to have the cell and nucleus membranes. Even the outer membrane operates as a highly active system. The amoeba extends tentacle-like arms in incessant search for food and strongholds, or footholds. It folds around outside materials and engulfs them. It contains many gateways where there's only selected molecules may pass. The two-way traffic shuttling through these gates is swift and steady. But the cell's food must come in through the membrane which surrounds it. This envelope has a marvelous capacity for letting in only what the cell wants and rejecting what it does not. It also has a remarkable flexibility which plays a vital part in many one-celled animals' hunting habits. Okay. The original membrane would have to form by pure chance. By pure chance, it would wrap itself around and seals within itself all the many complex and necessary cell parts that allow the cell not only to survive, but also to reproduce. Holes too small or too large in the membrane would be lethal to the newborn cell. Also selected gateways. In other words, yeah, it lets in amino acids, but only left-handed, not the right-handed. Otherwise, you end up with chaos and death. How did the nucleus membrane know to be so marvelously selective in getting all the chromosomes together into the penthouse of the cell? I mean, just the fact that it exists, why? Why would it exist at all? Evolution cannot learn from its mistakes. It can repeat the same mistake countless times, the definition of insanity, and yet it just happens. In other words, if these two amino acids don't go together, and so it comes together and they break apart. But the fact is that they're proximal to each other, and therefore they keep trying each other again and again and again, because there is no ability to understand. But what happens so often is that you have an implicit personification of evolution, that somehow it figures out, if this doesn't work, I don't try that possibility anymore. And so let me try something different, and let me try something else that's different. And then when I do have something that works, let me put it aside to safekeep it so that I can now build on it rather than that it falls apart in the time that it takes for me to assemble whatever, say for instance, the protein that it's working on. Not just to evolve the cell membrane, but to coordinate parallel evolutions and connect them at just the right moment at their random times when a viable cell can be formed. So in other words, all the other kinds of things, the ability to handle energy, the ability to digest food, the ability to get rid of stuff that's in the cell that is now waste, all of that stuff has to be there already when that membrane surrounds it, otherwise the cell dies. And now you start all over again. Energy conversion, ATP. Raw energy, sunlight, heat, electricity destroys and mutates, so very controlled use of energy converted into a usable form is required. Adenosine triphosphate, the standard energy currency of all life, to power biological functions, avoids deficient or excessive energy levels. In other words, what energy package it has is exactly what is needed in order to accomplish whatever the cell needs to be accomplished. It's not too much, it's not too little, it's exactly what is needed. Sugar energy is converted to usable ATP energy in 10 steps, but to net gain 2 ATP power pellets. 2 power pellets are first required to kick off the process, running at an energy deficit for 9 steps. Only in the last step are 4 pellets formed. In other words, if you have only a partial of this sequence evolved, you will kill the cell because it will run out of energy. It has to have all the steps there in order to accomplish what the cell needs in order just to survive, just to operate. It is purely wishful thinking to suppose that a series of 10 simultaneous beneficial additive mutations can produce 10 complex enzymes to work on 10 highly specific substances, and that these reactions would occur in sequence and all at the same time, and there it is. Okay, that's a fantasy, but it's a fantasy that seems to be stepped around because, well, we want evolution to work, right? As each cell's ATP molecules are used, unhooking the last phosphate, they are converted to ADP, diphosphate, and then are immediately recycled to ATP within 20 seconds. When twice as much energy is required, ATP simply unhooks 2 phosphates and then is restored in 2 stages. Mitochondria. Mitochondria are so elaborately designed that they have been referred to as cells within the cell. Their interiors look something like cutaway models of ocean liners, with many chambers and components and dividing walls. They are miniature power plants that provide the cell with the energy it needs for growth, reproduction, and other functions. The mitochondria have been described as cylindrical bodies and that is true if one thinks in terms of photographs which catch them in a fraction of a second. But in the living cells, they twist and slither along like eels, very peculiar eels, because they are constantly splitting into equally active bits and fusing end to end to form elongated swimming systems. One of the things that's fascinating is design, elaborately designed. But of course, that was an error on the writer's part. So you have at least a concept of what's going on in the cell. Similar size and shape as small bacteria, their own DNA similar to bacteria DNA ring structure, and it's theorized therefore that they were bacteria that were absorbed by the cell and have maintained some kind of integrity throughout the lifespan of all animals. Now the thing is that almost everything has mitochondria. At least all animals have mitochondria. It's not as if some do and some don't. And so if this is going to have happened, it would have to have happened at such an early stage for everything then to be able to have this kind of result. If so, then considerable autonomy for mitochondria. If they were already an existing self-handling object, then when they're in the cell, they should be beholden to nobody. Well, what do we find? Most of their proteins, even DNA synthesis enzymes, are coded by the nucleus genes. In other words, there is, of the human mitochondria's 83 proteins, only 13 are coded by the mtDNA, the mitochondrial DNA. Even then, large subunits are coded by nuclear DNA. If this is a pre-existing, self-existing object, then so much of it is dependent entirely upon the cell in which it resides, especially from the nucleus. Well, all proteins must be labeled and transferred across 2 membranes. How would these intact codes have gotten into the cell nucleus? Because the cell nucleus is set up to reject anything that might threaten the information that's there. So how did these things get into the cell, attach themselves to the chromosomes that are in there, and now become part of the functioning of the cell without any kind of a warning being raised? I mean, it's wonderful to postulate this, it's wonderful to imagine this, but the thing is that the practicality challenges the reality, or challenges the theory. Okay, conversion machine, ATP synthesis. ATP is produced in a tiny 100,000 would fit side by side in millimeter molecular rotary motor, rotating at up to 7,000 RPM, revolutions per minute. This is an engine, and this is the base of it. This is where you have the ATP, well, that's the ATP going out. You have the ADP, and you have the phosphates coming in, and it is creating at a rate of 70,000 RPM what the cells need in order to make the cells work properly. That's the base, and it's a motor that's driven by protons. This is in contrast to man-made electric motors which use electrons. These are the proteins. The protein goes in here, runs it around, and goes out there. And it is an engine that's run entirely by protons, rather, not proteins, protons. Here, adenosine diphosphate is combined with a phosphate ion in the presence of a catalyst to produce ATP, which is then released, making way for the next cycle. Okay, current of protons drop, let's see. Such nanomachine exhibits all the characteristics of self-intelligent, super-intelligent design. ATP is vital for life, and many of these motors were needed before the first living cell could exist, and especially replicate. An evolutionary possibility. In other words, for anything to happen, it needs to be driven by some kind of power source. But the power source has to exist already before those things could happen. So if there's going to be any kind of replication, if there's going to be any kind of digestion, if there's going to be any kind of building of protein, these already have to be working to provide the energy. And so now you end up with a catch-22. How can you have it and still not have it? The motor is present in all living cells. When it spins one way, it converts energy from digested food into the high-energy ATP, or it spins the other way, breaking down the ATP, making its energy available for use by other metabolic processes. This way, this is in, say for instance, you have the chloroplasts who create the ATP. And then it is now being broken down so that the cell can function. The chloroplasts are organelles of photosynthesis in higher plants similar to mitochondria, who also have their DNA, once again with a ring structure. According to the endosymbiotic hypothesis, supposedly the original cells started with anaerobic, without oxygen, without mitochondria or chloroplasts, then established a stable symbiotic relationship with the bacterium, subverting its oxygen, its phosphorus system to their own use. Plant and algal chloroplasts were derived from oxygen-evolving photosynthetic bacteria, repeated times from different pigments and properties found in the chloroplasts of present-day higher plants and algae. Fine print, this is only assumption and speculation. In other words, there's no way that you could possibly actually create this kind of a system actually happening. So it's wonderful that you can theorize it. Original cells started with an alternate energy conversion. In other words, where did they get their energy from it before they had the ability to have that ATP synthase motor? Who struck the bargain to not simply eat the bacteria? If criteria is, does it work, why aren't other methods still in the cell? Or are there cellular energy conversion efficiency audits done by whom? Cells are too clean. I mean, it should have lots and lots of other possibilities by which the cell would have gotten energy, but you don't have it. The ATP synthase molecule, the motor, is everywhere, in every living thing. What makes plants alone susceptible to this chlorophyll solution, when it may be advantageous for animals to directly make use of alternate power sources, especially when this supposedly has happened multiple times for plants. Okay. After the ATP, extraction of needed material from environment. In other words, how does a cell get what it needs when everything that it needs is out there? And so you have the membrane playing a very, very key role in ways by which it gets the food that the cell needs inside. Either it reaches out and encompasses the item and therefore begins to be eaten up, or that the item comes into the membrane and it just closes up the circle around it, or it goes and actually has little connectors that attach to it and then pull it inside in order to be digested properly or taken to wherever it needs the things to go. Ingesting the food from the environment, endo into cyto, cell, and oocyst, a process. Endocytosis is a process in which the cell takes in outside nutrients. How does it recognize food from non-food, or as in right-hand amino acids, lethal food? How does it know? How does it figure this out? How does it know when to open the door up for these nutrients to come in and otherwise keep the door closed? Osmotic pressure, the movement from a higher concentration toward a lower concentration, would prevent many nutrients from entering, since the cell would have greater concentration of proteins and so forth inside. And so you have the cell membrane holding these things, but at the same time being able to take what's out and bring it into an environment which should have a great amount. Say, for instance, if there's amino acid out there and it pulls the amino acid in, but the cell itself is filled with all kinds of amino acids. It isn't simply osmotic pressure. It isn't simply that the outside pushes it in. If anything, the reverse should happen. It should gut the cell from all that it contains. How can a cell digest food without destroying itself? How can the cell get the food where it is to be used and to the digestive processes? In other words, it's not just enough to get the food in, but where does the food have to go? How does it get there? You see, all of this has to be there already when the membrane first encompasses the cell if you're going to buy evolution. It has to have the ability to do all of this so that that cell can maintain its life. That you don't have food stacked up in a corner here, but it's not going anywhere. How does it get it to move? How does it get it to the places where it belongs? Garbage disposal. You know, something that's very basic as that. You have the proteasome. Proteasome breaks down the proteins of the cell's building blocks and many machines, identified by tiny protein garbage tag, ubiquitous. In the barrel, damaged, unwanted proteins are broken into reusable bits to make new proteins. You have the lysosome. Old and worn out organelles, you know, like mitochondria or whatever it may be, are placed into cellular stomach. An organelle called the lysosome. Its acid and several types of enzymes digest the unwanted organelle. This is called autophagy, Greek for self-eat. Bacteria, viruses, and other food the cell has engulfed, allowing the protein to be processed for reuse. Activating lysosomes protects mice from diseases like West Nile virus. Possibly this could treat diseases in which cellular trash piles up, including Alzheimer's and other diseases of aging. You have scrap heaps, like sweeping trash under the rug. Unwanted proteins may be piled in a gob called an aggregate to keep them isolated from the intense activity within the cell. The prion, this is the mad cow disease prion, protein forms aggregates which build up in the brain. In other words, it doesn't go anywhere, it just becomes where the brain cannot function, or the brain cells cannot function, because you have a buildup of more and more of the prion garbage. Extracellular, Some garbage is not digested by lysosomes, but may be spit outside the cell in a process called exocytosis. There, the trash may be taken apart by enzymes, or it may form a garbage heap called a plaque, which may still be harmful. In other words, some stuff it can't deal with, it throws it out. Goes into the garbage outside the cell. There it might be eaten up by enzymes out there. Or else, just like what happens in your blood vessels when a plaque buildup is created, and therefore your blood vessels are not as elastic, they begin to narrow, and then you have the makings for all kinds of cardiovascular disease. The cell may also pump out from inside what it considers toxic material. This may happen to cancer drugs meant to destroy cancer cells, even antibiotics rejected by their target bacteria. The task is to figure out ways to keep the medicines inside where they can do their job. So that the cell has many different options for getting rid of the garbage, or what it considers garbage, to keep itself alive and to keep the inside pure and clean. The DNA molecule, a remarkable evolutionary mystery. Okay, you read the letters there. That's just for starters. It takes 39,936 letters to describe the type, number, and order of chemicals making up the longest DNA sequence known to man. Brookhaven National Laboratory biologist John Dunn determined the sequence by analyzing T7, a bacteria-eating virus. Why all the fuss? Now that the sequence is known, biologists can take apart the virus DNA to study how it lives, dies, and mutates. The previous longest DNA molecule sequence was 16,569 letters long. So if you can imagine, you have just that as a starter for a DNA sequence. How do you put together 39,000, 40,000 amino acids? How do you build these kinds of things to make them work? By simple, pure chance. The famous double helix of DNA is like a spiral ladder in which nucleotide pairs of adenine and thymine, guanine and cytosine form the rungs. The order of these pairs is very precise. Total random connecting of the elements for DNA just will not do. Replication, cell growth, is tightly controlled by certain protein molecules, enzymes, and RNA, which in turn can only be produced at the direction of DNA. So how does it all happen when you have it so tightly controlled and circular? The DNA creates an RNA that then now builds the DNA that creates, it's all a circular pattern. It isn't as if something that exists outside and now creates inside the cell what it needs. It is that the cell is able to do this all on its own. Did the DNA code and the means to translate it appear simultaneously? Incredible for any such coincidence, given the extraordinary complexities. And we'll get into that with the next topic, with DNA. And there is such a fascinating area in that. Something that supposedly happened by chance should have as its product something that leaves nothing to chance. Amazing that if it is something by chance makes it, you would think that that's the healthy way the thing should go. Not that it so locks it down so that the replication of the DNA is very, very guarded. Not to say that things don't happen, because in this world, yeah, you do. But the thing is that when you consider how many millions of times you have this replication going on in your body all the time, and to have it so that it stays consistent, so that you don't have things like cancer happening all the time in all parts of your body. Mitosis, meiosis, the amazing ability of the cell to replicate its DNA. When you take a look at this, this is amazing. You have the first where you have the two centrosomes, and they are now beginning to form. And meanwhile, the chromatin, which is the DNA in the nucleus, begins to start forming into chromosomes, and then they start attaching to each other, and then you have, now that they are all attached to these microtubules, and you have little engines that now will hold them to the different centrosomes, like you have here. And you have this extraordinary, ordered, extraordinary, complex, and yet efficient way of creating cell division. And then here you got the membrane again, the cell membrane, and it knows where to divide the cell, so that you have both sides of the cell have equal chance to survive. And what you have here are the actual pictures, at least, of this. So you start out with each of the different ways by which you actually can see where the chromosomes are getting to the point where they're now in place, and they start to get pulled apart or pulled together or bolted to their different poles. A highly non-random process. It isn't that some do and some don't, some chromosomes are and others aren't, or maybe you'll have maybe about 6 chromosomes on one side and 20 on the other side, or anything else like this. A highly precise replication, highly regulated phase order, distinctive tasks for egg and sperm division. In other words, not only do you have the division of a cell, but now you have highly specialized so that, for instance, sexual reproduction, you have the egg and the sperm the way it needs to be in order to form a future, well, animal, human, whatever. The whole cell and nothing but the cell. Life is more than all of its component parts. You can find all the components of life in the corpse, but it is not life. Remember Polanyi's irreducible structures. Just because you have the parts, it does not mean that the controlling ability is going to be created by those parts. The next level up has to exist for the lower levels to operate the way they're supposed to. A highly structured lab process to obtain the components of life indicates that life is an undertaking that requires a designer or an organizer. Yeah, it's wonderful to say, oh yeah, we created amino acids or anything else like that in the laboratory. But when you realize the extraordinarily elaborate material that has to be, equipment that has to be used to have that happen, and this is something that the cell is doing constantly, always, in your body. It doesn't need all of this other stuff. And somehow it knows everything it needs to know in order to do the job. And interesting too is that so often with these lab experiments is that they will start off with purified components from an industrial chemical warehouse, not using the dirty chemical environment of naturalistic chemistry. In other words, already they're identifying the fact that you need to have something very, very special even to achieve what they want to achieve. So yeah, they have amino acids and they create amino acids, but they're left and right-handed. And so they go to the industrial chemical warehouse and they get the pure form of the left-handed. And now they have these amino acids, and then they create the next step. But the next step doesn't necessarily mean that the right and left-handed are going to be the way that it's supposed to be. And so they have to go back to the chemical warehouse and get purified form for the next step. And all of this that they are admitting needs some kind of intelligent oversight in order for them to achieve what they want. Too much has happened all at once for the first cell to take shape. Everything is supposed to be random. A workable solution at this moment may not exist in the next moment. Remember that what can connect two compounds together can easily destroy that connection. So the enzymes and the catalysts can do just as much damage as they can in creating something. As a matter of fact, that's one of the problems that so often happens, is that once a catalyst has reached a certain point, then it starts trying to take apart everything that it's been doing. A good DNA sequence would have no mechanism to save its effort. How does it know? How does it know how? How does it know where? How does it know how to safeguard it? How does it know to be able to accurately replicate it? How does it have all of the things that it needs already in place in order for that to happen? And you're saying this has happened completely by chance. A lot of nonsense might form, duplicate repeatedly, just as easily as useful material. A cell based on mere chemical reaction composed of chance connections would preserve everything or nothing, having no ability to choose apparent worth. And yet there is no apparent trace of anything useless in any class of living cell. Evolution makes a good story, but isn't reality. A tremendous amount of elements, compounds, functions would have to be enclosed at once at the first cell membrane wraps them up into the first cell. So much of it defies credibility. Multicellular cell specialization, multicelled organism, all cells originate from a single cell. Cells communicate, work together to support each other, although the cells maintain their full range of life tasks. Yet when removed from the body, cells and organs will die unless special supports are in place. In other words, they have become so the cells are so interdependent that they cannot operate by themselves at all. The cells permanently turn off survival equipment to depend on other cells. Therefore, they will die if any other tissue class ceases to function properly. So when the kidneys go, you die. When the heart goes, you die. When your lungs don't work, you die. The whole organism dies. That there is nothing that will step in to take its place. Fully altruistic, it does a function, like for instance, lungs, or makes a substance, for instance, the endocrine glands, for cells for which it has no contact. It isn't as if they're next to each other and therefore one can share the other. It is that you have different tissue in the body that has no real direct relationship. The lungs do not have direct relationship to your toe. And yet what the lung provides is necessary for that survival of that toe. How would cells randomly develop the mechanism to activate for the life of each cell this only one function and then congregate with only like function cells? Useless evolutionary dead ends should certainly be expected. Yet the lack of vestigial structures identifies a tight-knit interaction of all cells in the body. It seems to require a sentience on part of the cell to affect this kind of coordination. Very difficult to maintain evolution's non-directed, no-goal oriented nature. Serial cell differentiation. So you start out with stem cells. They're self-renewing. The daughter cells exactly like the mother cell. Relatively, these are very few in number. Some of the daughters go into what's called transient amplifying cells. They multiply, they begin to shift toward specialized cell lines. For instance, blood cells. And then these daughters, when multiplied, produce mature specialized cell lines, erythrocytes and leukocytes and thrombocytes. These cells do not divide or differentiate. In other words, when you get to the end product, that's it. That's as far as it goes. When finished, they are deleted and their components are recycled. The process reduces cell division frequency at a basic level, reducing permanent changes. In other words, you have very few of the stem cells replicating. So you only have, say, for instance, one daughter. That one daughter may go through, say, for instance, 5 divisions. Now you have 32 cells. And of those daughter cells, maybe they go through 5 more divisions. Now you have 1,024. And so what you have is a very efficient way of maintaining less and less problems creeping in because of duplication. That you have less and less potential of some kind of mutation taking over, because the ones that create all the others, these duplicate very, very slowly. And yet, by the time that you finish, you have an enormous amount. So for instance, for your skin cells, which are constantly renewing on the outside. But the thing is that because you have these steps that are in the way, you can have the full supply that you need for your body, and yet still not create so many possibilities for mutation to enter in, or some kind of error to enter in that then affects everything else. Enormous stability and resistance to change, even if some cells become more fit, reducing their ability to take over the organism and disrupt bodily integrity. So that's one of the problems with cancer, is that they don't die the way they're supposed to die. And they don't stop producing or duplicating the way they're supposed to. And so they do take over the organism. But the fact that you don't have cancer happening that frequently or all over the body in many different functions and many different tissues indicates that this kind of a thing is very, very good in terms of stability. Catch-22, multicellular organisms could not emerge before organism-level selection had left to the evolution of mechanisms to suppress cell growth. In other words, you have organism level suppresses the cell level selection, and yet the cell level selection had to be there in order to, according to evolution, be able to have the next higher levels appear. Okay, apoptosis, Greek for falling away. This is kind of an interesting, and has become something that has become more and more a topic of investigation over the last 20 years. Up to that point, nobody really seemed to really bother too much with it. And now you find there's an awful lot of interest in what is going on here in terms of the cell. Getting a hand. At first the hand is a bud, then a paddle, and then as the fingers become more articulated, the tissue between them falls away. An inbuilt cellular program terminates the existence of cells after a predetermined period. All cells eventually end in time. Some in about 120 days, for instance, red blood cells, others in 120 years, like the pluripotent stem cells. In other words, the original stem cells that were right at the top of all the rest, that they too have a clicking time watch, time bomb, if you will. The current approximate upper level for human life is 120 years. Entirely natural, physiological process, essential in an organism's orderly life. So for instance, post-wound repair, you have cells that are being created to help repair that wound. Or you have menstruation or lactation, and you have changes that happen once now this particular function isn't necessary anymore. So in other words, when the placenta has grown and is no longer needed, you have a programmed cell death or lactation. Once that the woman has passed, say for instance, the woman has passed nursing her child, there are cell changes and there are some cells that now disappear, as well as its development. In other words, caterpillar to butterfly, you have this cell death, or tadpole to frog as it loses its tail. That you have either the orderly life or the development. You have this occurring. This idea of apoptosis. Apoptosis versus necrosis. Necrosis is catastrophic death. Results in trauma, toxins, etc. Results in rupture, disintegration, debris, decay. In other words, the cell just literally falls apart. Its cell membrane just disintegrates, and everything leaks out. For apoptosis, it's a controlled disintegration within life. So you have genetic control. You have signaling that involves proteins and RNA synthesis. In other words, you have proteins that are created in order to make this happen. It isn't just simply that everything falls apart. It is that these proteins go in there and they now govern what is happening to that particular cell. Packets of functioning organelles encased in membrane to be assimilated to other cells. In other words, here everything just leaks out. Here what you have is that you have packets that are enclosed in cell membrane. And so you may have mitochondria, you may have some chromatin, you may have some other kinds of things that now exist, and exist in a sense of still living, then which are then absorbed then into other cells, and they now become part of what that cell contains. Cells can simply disappear within an hour without a trace left behind. It's something that can happen so very, very rapidly. And yet, it's all very controlled. It's programmed, energy-dependent, cascade of biochemical and molecular changes in the cell. Mitochondria release certain molecules into the cytoplasm to help execute apoptosis. All components must be present, an insurmountable barrier to evolutionist explanations. In other words, you can't have partial. It can't be something that, well, part of the way, but it hasn't fully been developed yet, and so you only have half of the way. No. Sensitivity is such that the slightest change in a single protein derails the process. A powerful argument against step-by-step evolutionary development. Apoptotic pathways have all the hallmarks of irreducible complexity. Something is very, very strongly in charge as this is happening. It's not just simply that cells fall apart. And when you have a problem with it, it's what's called dysregulation. That's when you start having aging, cancer, AIDS, Alzheimer's, rheumatoid arthritis, embryonic lethality, eye problems. These all are affected because somewhere the apoptosis has gone wrong and has now therefore either destroyed something or has not killed. It has let grow, like for instance with cancer. It has let something develop and multiply that really should not. So that something has gone wrong with the ability of apoptosis in order to fulfill its task. Very, very, very interesting. The chaplain sure looks holy sitting there in that sunbeam. Yep. And as I walk outside, it's all raining on that. When you talk about what the Lord can do inside the cell, there's such in contrast to what is out there in nature by itself, that the cell is indeed a most marvelous, marvelous creation by God. Okay, assignments. Science versus evolution. We got 2 chapters here, total of about 49 pages, 50 pages. 2 short online articles, and then that you come up with some kind of a way by which, how are you going to express something that was talked about in this particular topic to somebody else. Thank you, and we'll see you next time in regard to the fascinating topic of DNA.